Your body's cells are diploid. That means they have two copies of each chromosome. There are 23 human chromosomes. And when a sperm with 23 chromosomes fuses with an ovum with 23 chromosomes, the result is now a new cell, a zygote, which has two sets of 23, 46 chromosomes. Now, when cells divide, the challenge then is that all of the daughter cells, generation after generation, have to have this same number of chromosomes. So let's imagine for a second that here is that zygote with its 46 chromosomes. If it didn't replicate its DNA, then when it divides to make two cells, then each of the two cells would then have 23 chromosomes. When each of those cells then replicated to make four cells, then the total would be about 11 chromosomes per cell. When the cells divided again, it would go down by half. When the cells divided again, it would go down by half, and so forth. So you couldn't get past an embryo, which was a microscopic ball of cells, before you got to the point where each cell no longer had a full chromosome. But cells need far more than full chromosomes. What they need is the entire set of 46 chromosomes, because then if they have 23 chromosomes, they have all of the human genes. If they have two sets, then they're expressing appropriate levels of all of those genes. And so if you were to go into a cheek cell, it has 46 chromosomes, the same 46 as were in that zygote. If you were to go into a bone cell, it has 46 chromosomes, the same which were in those that zygote. And the reason for that is that as a cell is preparing to divide, it goes through this S phase where the DNA is replicated. And so there are 46 chromosomes here, but by the end of S, there are two sets of 46 chromosomes, which are now adhering together thanks to centromeres and the cohesins, which bind them together. When a cell then finishes its mitosis, then that parent cell, which started off with 46 chromosomes, can then divide to make two daughter cells, each which has 46 chromosomes. And so thus, your brain cells have the same genes as your smooth muscle cells, or your bone cells, etc. How does this occur at the molecular level? Well, this is what we refer to as DNA replication. And it was originally described in prokaryotes, in bacteria, where the process is much simpler. So I will refer, sometimes to this is what happens in prokaryotes, eukaryotes. I remember because, I'm old. When I was in school, very often the process could be described in prokaryotes only. And it was only subsequently that, and we now know that in eukaryotes it happens this way. So in the S phase, how is the genome duplicated so that when a cell divides into two, there are now equivalent chromosomes for each of the two daughter cells? Well, first, you have to know where to start. Where do you start this replication of the DNA molecule? Replication doesn't occur any old place. There are specific sequences called the origin of replication. Now, if a simple bacteria like E. coli just has one circular chromosome, that means that there is then one origin, one region that has, say, 250 base pairs, give or take. And from that spot, then replication begins and the entire chromosome is replicated from there. In eukaryotes, however, the areas are bigger, and then an area which is replicated from that origin is what's called a replicon. So eukaryotes having so much more DNA can't get by with simply one origin of replication or even one per chromosome. So in humans, there are multiple origins of replication on a single chromosome. And so here's an origin, a little farther on, there's another origin, and the area which is replicated from one origin is what's called the replicon, which can vary between 40,000 and 100,000 base pairs. Now, human cells can sometimes vary in how long it takes them to replicate their DNA, because not all origins of replication have to be recognized. So an embryonic cell, which is dividing more quickly, could then utilize all origins of replication, while later in life a subset could be utilized. There are many proteins which bind to DNA and specific sequences of DNA, and there are then proteins which bind to the sequence of DNA at this origin. And so the origin recognition complex then is a set of proteins all coalescing on the origin of replication. This in eukaryotes then include some of the cyclin-dependent kinases and proteins made briefly at the end of G1. So we control our cell division. Cells don't divide any old time, and certainly we want to protect against cancer. And so if you need specific proteins in order for the origin of replication to be recognized, one could only, express those proteins at a specific stage of the cell cycle. At the end of G1, for example, before entering into the S phase where the DNA is replicated. So it is now from this spot, once the origin of replication forms, that the double helix of DNA then unzips to create single-stranded molecules, which can then be copied. Now, something I don't add here, really, because that would just complicate matters. Single-stranded DNA can fold upon itself and find homologous areas. And so another type of protein which we can find throughout is what are called single-stranded binding proteins, which then bind to the DNA and prevent it from folding in ways that we don't want. From this origin, then, enzymes known as helicases will then move in opposite directions. They essentially unzip this double helix to make that single-stranded DNA. So because we have replication forks now going in opposite directions, there would now be two different helicase molecules moving in opposite directions. A problem if you're going to unzip, if you're going to unzip a coiled molecule, as DNA is, then that would start to provide, kinks in the chain, what's called supercoiling. So in addition to helicase, which is here in blue, we would then have proteins traveling with the helicase, known as topoisomerases, which will then prevent this supercoiling. Because in this animation, the strands are laying flat, and you can unzip them and they just kind of stay laying flat. But in 3 dimensions, that's not what happens. These are coiled molecules. And if you try to separate a coiled molecule, the ends start whipping around. In a microscopic cell, you can't have that. And so therefore, what's happening is the topoisomerases can actually cut DNA and then pass one strand under the other and then seal it back again. And that will either lessen the tension, make less supercoiling, or to recuperate the original state, add more coiling. And so this is a coiled molecule, this double helix, and we don't want it to become too coiled. Otherwise, as we replicate it, the ends whip around. Or we want it to return to that original state of coiling. So the topoisomerases, following the replication fork, they can make cuts in the DNA and have one strand pass underneath the others. So here in yellow, we see the topoisomerases, and here we see we are unzipping the DNA as we go. And so this is going in opposite directions. So we have one replication fork moving one direction, another replication fork moving in another direction. One of the things that, once again, this animation isn't true to life, is that lots of these proteins, I'm having them as different, colors and having them as separate. What actually can happen is they form one multi-protein complex, and the DNA strands can kind of wrap around this as they go. It's actually quite complicated. And I could have something there that, like a big yellow blob or a big red blob. But actually what could happen is that different genes encode for different subunits, which then kind of coalesce. So it's actually more complicated. Okay, so now we have single-stranded DNA, and we are ready to make the complementary strand. Now, since we have DNA and we want more DNA, one would think then that we would start putting new DNA nucleotides, and the DNA nucleotides have the red sugar ribose, the red pentose sugar. So you'd think you'd want to add them, but actually you see that we're not incorporating into this area called the primosome, all of these proteins stuck together. If you notice then, we have ribose in the nucleotides which are coming in, and in addition we have uracil, which in this animation is white, as opposed to thymine in the DNA, which is blue. So here, as we go along, it is RNA nucleotides which are being added to form a short RNA primer. So we're going to begin the process of making new DNA by making RNA. Why? Well, two reasons. One is the physical. This enzyme here is an RNA polymerase adding RNA nucleotides. There are DNA polymerases, but they can't start from scratch. So DNA polymerase could start once there was something to add onto. So now that we have an RNA primer, DNA polymerase can now add on additional nucleotides. But DNA polymerase could not start from nothing. And so therefore, if we're going to start from nothing when we first unzip the molecule, we need an RNA polymerase. Why can't DNA polymerase do that? Well, the simple answer is because you don't want it to. And the reason for that is when you start to make a complementary strand, it turns out that the beginning of this process is the most error-prone part of the process, where you're most likely to make mistakes. You don't want then to have more mutations than you're already going to get. And so as a result, the very first nucleotides which are replaced are in the form of an RNA primer, which will eventually then be removed and replaced. So the fact that it was the most error-prone does not matter because these will not stay. Okay. And so if we are going to start to make a new DNA strand, we need to first start with an RNA strand to make an RNA primer. So DNA polymerase can't just start adding new nucleotides here. That's why we need RNA nucleotides. And notice the sugar here is in green, as opposed, so that represents ribose, as opposed to the deoxyribose here in red. And also notice that the RNA primer includes uracil binding to adenine. Adenine is pink, uracil is white, as opposed to thymine, which was in the DNA chain. Now, one other thing that I will repeat, I'm sure a lot of students are familiar with, early biology classes with ATP. ATP is adenosine triphosphate. There's an adenine, a ribose, and then 3 phosphates. And these phosphates store high energy in their high energy bonds, and we can use that for muscle contractions, etc. Well, not only do we have ATP, but we also have GTP, CTP, TTP, and this can be both in using RNA nucleotides and DNA nucleotides. So these extra phosphates store energy. And notice that all of the nucleotide precursors are in the triphosphate form. And when you want to add them to a new chain, you remove the phosphates and utilize the potential energy which was stored there in order to make the new bond. Okay, so we now have RNA nucleotides composing a short RNA primer. It can vary from organism to organism, maybe 6 to 9 nucleotides, which are then used. And now we can start to add the DNA nucleotides. Now, one thing which is a little tricky, DNA nucleotides can only be added in the 5' to 3' direction. So when a new nucleotide comes in, it has to come in with the 5' end. That's the end that has the phosphates. It has to come in with that facing the old nucleotide to which it's being added. Okay, so notice that we made a bond using the 5. This is the 5' carbon. Here's the 3' carbon. The new nucleotide is going to come in so that its 5' end can be attached to the 3' end of the original. So that comes into play in just a minute in the next video. New nucleotides can only be added when the 5' end comes in first. DNA polymerase adds new nucleotides from the 5' to 3' direction. Well, not only is that a mechanical issue that DNA polymerase has to do that, but then secondly, that's how we want it to work. And here's the reason. Let's say that I am about to attach this thymine as the complementary base pair to this guanine. Now that's incorrect, but let's say that I'm about to do that, all right? Well, it will bulge because it doesn't fit really well, and I'll recognize that. We have DNA repair mechanisms, which we'll talk about later, which would say, wait a minute, there's a distortion here. This isn't the correct pair. We need to remove that and try again. Well, we can. And the reason that we can is because the next nucleotide we try with is going to come in with those phosphates and those high energy bonds facing the place where they need to attach. So we could try again. But let's say we make a mistake again. Fine, there's a distortion. We recognize it's a mistake. We remove it, all right? And then we then try another time. And because this new nucleotide comes in with the appropriate triphosphate bonds, we have energy in which to make the correct correct bond. So we can do this all day as long as it takes to get the correct pairing. So because each nucleotide comes in with its high energy bond intact, so that we have energy to make a new bond, we can fix mistakes. If DNA polymerase was replicating in the opposite direction, all right, so let's say it was replicating this way. It doesn't, but so let's say it did. If the incorrect nucleotide was coming in with its 3' end first, and here's the existing one we're adding it onto. If we then remove those 2 phosphates, all right, if we made a mistake, if that was the wrong nucleotide and now you wanted to add the right one, the problem would be there is no longer any high energy bond here. So you would not be able to fix mistakes. Once you made a mistake, you would not be able to correct it. No new bond could form. And so DNA polymerase can only add new nucleotides in the 5' to 3' direction. So that means if we were to talk about the replication of DNA, we would then have to distinguish between what's called a leading strand and a lagging strand. So first we unzip the DNA using the helicase and the topoisomerases. The primase would then begin to add the RNA molecules, the RNA nucleotides of the RNA primer, all right? And once the primer was made, we could now start to add DNA nucleotides, right? So notice that there are 8 RNA nucleotides forming this RNA primer before the first DNA nucleotide can be added, so that RNA primer is what DNA polymerase then attaches the new nucleotides to. DNA polymerase could not start from scratch without that RNA primer. Now, one replication fork is going this way. And then over here, the other replication fork is going this way. Now, notice as the replication fork just unzips and unzips and unzips, this is convenient because the next nucleotide comes in with its 5' carbon first with those high energy bonds. Same thing here. If we want to add a new nucleotide here, then the next one will come in with its 5' end first. And then we can just keep on going and going and going and going and going, because every new nucleotide comes in with the appropriate end first, that 5' end, and we can then attach the new nucleotide to the old one which was already there. And this can now just continue as the replication forks continue to unzip and then go towards the end of the cells, or until we meet the next replicon. And so this is what's called the leading strand. It can just keep on going. We only have to start it once. Once we start unzipping and we make one RNA primer, we can just keep going and going and going and follow it as long as we need to. And so this is what happens on what is called the leading strand. But notice that this area has not yet been included and this area has not yet been included because one cannot follow the replication fork on that opposite end and be going from the 5' to 3' direction. So that's what's called a lagging strand. So there's a problem. So if we were to replicate this top strand here, starting from this point moving to the right, we could just keep on going and we can add new nucleotides in that appropriate 5' to 3' direction. But that is not the case here, all right? So here, notice that so let's just talk about this strand right here on the right, on the bottom. The replication fork is moving farther to the right, but the 5' to 3' direction means that from this point here, we're now going to move to the left if new nucleotides come in with their 5' end. So notice the 5' end would attach here, all right? If we had a new nucleotide coming in this way, it would be attaching with its 3' end. That's the wrong way. And so we make one primer here. And once we get to 8 nucleotides, now we can add a DNA nucleotide, and now we could keep on going in this direction. But by that time, we've unzipped a little more. So we could now make a second primer. A second primer of RNA nucleotides, to which then we can add DNA nucleotides going from here to where the first primer started. So here we are replicating the bottom strand, or on this part, the same thing is happening on the upper strand. Notice there is now new DNA being added, and we will keep on adding it until we get to this portion here. However, this is not being made continuously. The leading strand is replicated continuously. This is now being made. Here was the first piece. Here's a second piece. And now if we added a third primer, we could now add more DNA. So here's new DNA nucleotides coming, which will then join our second primer. And the second thing is going on up here. So moving to the right, this is what's called the lagging strand. This one up top was the leading strand. However, on the left, This bottom one was the leading strand. This is the lagging strand. The lagging strand must be made in a series of fragments which get joined later. And so as we unzip and follow the replication fork, one of the two strands can then be copied continuously. That is the leading strand. And then the other strand has to be made in a series of fragments. So notice that, here's a piece, there would be another piece there. Okay. Now, there are different DNA polymerases. I'll use the numbers from bacteria, which are simpler. So while DNA polymerase 3 is the one that was adding new DNA nucleotides, DNA polymerase 1 is going to have a role in just a second. In eukaryotes like ourselves, it's more complicated. There's more options. So let's just stick with this one. So DNA polymerase 1, what it's now going to do is to replace the RNA nucleotides with the green pentosugar ribose and replace it now with DNA nucleotides. And so while it was great that we made these RNA primers, if we're going to have a DNA molecule, all those nucleotides with the green, they then have to go. And so a different DNA polymerase is going to remove the RNA primers and then replace it with DNA nucleotides. And then another enzyme called ligase will take the spot where the last primer, which has now been replaced with DNA nucleotides, where that just ended, to seal the nick, to add, to connect that last nucleotide of the replaced primer to the original set of DNA nucleotides, which was already there. So this would happen in bacteria once and replicate the entire chromosome, or in eukaryotes, this would happen in a region of a chromosome called a replicon. And once all of the different replicons were replicated, then we would now have two copies of this chromosome. So here is a chromosome. A chromosome is one really long piece of double-stranded DNA. So the chromosome 1 you inherited from your mother looks like this. It's one really double-stranded molecule. The chromosome 1 you got from your father, the same thing. But before a cell divides, you want both daughter cells to get a copy of the chromosome 1 you got from your mother. So if this is the chromosome 1 you got from your mother, we have to replicate it before you have two, which you could then give to each of the two daughter cells. So we find an origin of replication. Helicase unzips the DNA as we proceed. Topoisomerases is adding coils so that we don't have supercoiling and we have the right degree of coiling. We need to add RNA primers at first. So the enzyme called primase will make a short RNA primer of, say, give or take 8 nucleotides. And once that happens, then DNA polymerase 3, which once again is a complicated enzyme that has lots of little pieces. I'll just make it an orange blob, but it's far more complex than that. It now starts to add new nucleotides. The leading strand can simply have more and more nucleotides added in the same direction as the replication fork is proceeding. While the second strand, what's known as the lagging strand, has to be made in a series of fragments. And so we'll have an RNA primer, a fragment of DNA, an RNA primer, a fragment of DNA, an RNA primer, a fragment of DNA. These are known as Okazaki fragments. Then another DNA polymerase 1, will replace the RNA nucleotides of the primers with DNA nucleotides, and then another enzyme called ligase will then seal the last nick connecting the last nucleotide, which replaced a primer nucleotide with the DNA of the existing strand. So this is what's going on in the S phase. Look, we now have 2 copies of the chromosome 1 that you inherited from your mother. They will stay attached to each other at a centromere, but also there are proteins called cohesins which will hold these 2 together until it's anaphase, when the cohesins get cut, and then you can pull this one to one daughter cell and this one to another daughter cell, so that each daughter cell now has a copy of the chromosome 1 you got from your mother. In that second, we have upwards towards 10 million cell divisions in our bodies, for our red blood cells, our white blood cells, our skin cells. We have lots of cell division, and certainly it took lots of cell division to turn you from that microscopic embryo that you once were to what you see when you look in the mirror. And every cell division then needs an S phase, and then this DNA replication then occurs with all of your nucleotides in that S phase. Now, look at all of these nucleotides. That looks like a lot, but your genome is more than 3 billion nucleotides. So this is certainly an elaborate endeavor. And when you think of all of those triphosphate bonds to make new nucleotides, it's also a costly endeavor. In my genetics classes, I like to talk about telomerase later in the course, after the unit where I talk about DNA replication. The problem is it's also relevant here, so I'd like to just mention it kind of quickly. I have a video or 2 on it which go through it to greater degree, so I'll mention it here, but once again we can get to it later. So as I had mentioned, these RNA primers are there so that DNA polymerase has something to stick its new nucleotides to, and we don't worry because we can remove the primers later and then add new DNA nucleotides. And that works, except at the end of a chromosome, right? So here we are at the end of the chromosome. If we remove the primer, all right, and we say, oh, we'd like to now add new DNA nucleotides. The problem is on one of the strands we can, all right, but on the other strand we can't, because the new nucleotides would have to come in on this strand with their 3' end facing in, not the 5' end, right? The new one coming in would come in with its 3' end first. So that's potentially a problem, where that means that this chromosome would stop. We wouldn't be able to replicate this part. Thus, it would be a little shorter than it originally was intended to be, because we can't replace that. Well, you're thinking, that's not a whole lot of nucleotides, right? But then the next time we replicate this cell, the chromosome would be still a little bit shorter, and then it would still be a little bit shorter, and then a little bit shorter. The chromosomes would shorten every cell division, and there would come a point where the chromosome was too short then to replicate, and a cell would be done. It would say, I'm not dividing anymore. I have reached the end of my ability to divide. Well, to prevent that, there are regions of the chromosomes called telomeres, which contain a repetition of short base sequences. So telomeres in humans, somewhere between 250 and 100,000 times, repeating this sequence. And what can then happen here is proteins bind to the telomeres, and now RNA molecules could actually be used as a template to make the DNA molecules to fill this space. Now, this is weird in that usually DNA serves as the template for RNA. That's what we call transcription. But here we have an RNA molecule incorporated in this enzyme telomerase. It is the RNA molecule which is serving as the precursor for the DNA nucleotides to be added here. This is reverse transcriptase. And so here you have this weird enzyme that has both RNA and protein in it. That thing is called a ribonucleoprotein, and it then serves as the template to make sure that the ends of the chromosomes can be replicated. So there's that. Let me just say one final thing. Telomerase is fascinating because you say, oh, I'm so grateful I have telomerase. Now I can replicate the ends of my chromosomes. My chromosomes won't get shorter with each cell division, ultimately getting to the point where they can't divide. That's good. And that is good in what's called our germline, all right, because you don't want, with every generation of humans, chromosomes to get shorter, because otherwise at some point, the human species would end. We wouldn't have enough DNA if the chromosomes keep getting shorter and shorter. So in our germline, our chromosomes stay the same length. Interestingly, however, in much of our bodies, we turn telomerase off. So we can extend the lengths of the chromosomes at the ends. We choose not to. Why? Well, this is actually one of the reasons that we get old. As we get old, we start to break down. When cells die, they can't be replaced, all right? And you say, why? Why would we want that? Why would we want to have cells have their chromosomes get shorter and shorter to the point where they couldn't divide anymore? And thus, as we age, we're less able to repair our tissues. Why would we want that? And the answer is that's one of our ways of fighting cancer. No cell then, or most cells, are not immortal in that they have a certain number of cell divisions and then they're done. Their chromosomes are too short. And so therefore, if a cell is making a mistake, if it's dividing inappropriately, in most or in many cases, then it can only do so for so long and then it's done. It's a way of protecting ourselves from cancer. So while we're afraid of aging, we're also afraid of cancer. And how do you balance the two? Well, why do we have cancer? Many cancer cells actually, they have mutations. That's why they're cancerous. One of the common mutations is to express more telomerase, so that a cell which shouldn't be making telomerase now is. And so therefore, it doesn't have that shortening, and it is now immortal. It can divide and divide and divide with no end in sight. So just talking about DNA replication, there is that special consideration at the telomeres, which are the ends of the chromosomes.